I'm here at the second European Barcoding Conference. It's part of the 2010 International Year of Biodiversity. And one of the extraordinary things about barcoding is its ability to discover a new species that we haven't found before. And I'm with somebody who has not found one or a few, but hundreds of new species of small gastropods on the islands of Vanuatu in the southwest Pacific.
My name is Sophie Van Malen. I've been always interested in mollusks since I was little, and I had the opportunity to do my master thesis with Philippe Boucher at the Muséum National d'Histoire Naturelle à Paris. And he gave me the samples they collected on the Center 2006 expedition in Vanuatu. Work on triforids, which are very small micro mollusks. Small, it means like it ranges from 3 millimeters, sometimes even smaller, to at most 2 centimeters. We had a lot of specimens. We had 23,000 specimens in total, which contained both dead material, empty shells, and live specimens. The live specimens for barcoding were only represented by 525 specimens, and we divided them into morphospecies. It corresponds to 73 morphospecies. All of them are new, actually. And so in total, the 23,000 specimens we had, they correspond to 257 morphospecies, of which maybe 1 or 2 or 3 are known, and all the rest are probably all new species.
So you're telling me that essentially all of the species that you found are new to science? You know nothing like this from anywhere else in the Pacific?
We know in the Pacific there are different places where there is at least this much diversity, even more. And these micromollusks, because they are so small, it's very hard to sample them. It's also very hard to collect them, to handle them for sequencing. Therefore, there's not a lot of people who work on them. And therefore, actually, the largest part is not known for science. It's all new. To give you a comparison, triforids is one of the 5 most diverse families of mollusks, and there is no sequence in GenBank.
This is absolutely fascinating. Thank you so much for talking to me today. To find out more about barcoding, I talked to David Schindel, who is the Executive Secretary for the Consortium for the Barcode of Life, funded by the Sloan Foundation and hosted by the National Museum of Natural History.
Well, perhaps this is apocryphal. I don't believe it is. I believe it's a genuine story. Paul Hébert, this Canadian geneticist, was in a supermarket looking around, probably standing in line waiting for the cashier, and he wondered how many products are in this store, given that they can all be identified by the scanner that's using an 11 or 12 digit code, the Universal Product Code. Well, the typical supermarket has 30 to 40,000 specimens, that is, species of product, distinguished by 12 digits. Now there's 2 million species known on Earth, figure 20 million or even 200 million. The combinatorial mathematics says that 500 to 1,000 base pairs of DNA using just the third position, the silent third position, is way more combinations than you need to diagnose all the species on Earth, and that's a very powerful idea.
Hi, I'm Bob Hanner. I'm the Associate Director of the Canadian Barcode of Life Network at the University of Guelph in Ontario, Canada.
One of the things that intrigues me, because as a conservation biologist I want to make sure that I'm eating sustainable fish, is the reality is that I'm often not. Tell me about what you've done to throw light on what the fish are that we eat when we're being served cod or sole or red snapper in a restaurant.
Well, this is a really interesting question. Working with a team of international scientists, over 200 ichthyologists and geneticists from around the world, we've been compiling a reference sequence library of DNA barcodes that can uniquely identify the fish species on our planet. So far, we've barcoded over 50,000 specimens and have developed a reference library for nearly 8,000 species. And one of the things that we sought to test was whether or not our database was robust enough to identify the species that we encounter in the marketplace. And so a graduate student and I and high school students in the United States and Canada began collecting fish samples from the markets and barcoding them, and we could identify them to species in all cases, but what was shocking was that 1 in 4 fillets that we tested was not what it was labeled as, was that this is a systemic problem, and it happens in many different species. We're often being served tilapia in the place of cod, or hake in the place of cod. And those kinds of things constitute market fraud, and nobody likes to be ripped off. But there are more insidious problems that we've seen that relate to environmental sustainability, as you've alluded to, Stuart. One case in point is the Atlantic halibut fishery is nearly collapsed, and they're considered a threatened species. Pacific halibut, which is a different species with a distinct barcode, has been more sustainably managed and is considered an eco-friendly substitute for Atlantic halibut. And in some cases, we were finding so-called Pacific halibut actually barcoding as Atlantic halibut. So even when consumers are trying to make an ethical choice, it's being undermined by this problem of market substitution. And for the first time, the DNA barcode library we've created through Fishbowl is allowing us to address these kinds of problematic problems, problematic processes.